So next up we have Ivan Klbik talking about the post-hypertonic lysis of red blood cells and cell volume regulation.
Thank you for introduction. Today I would like to tell you something about research that I have done about post-hypertonic lysis of red blood cells. Post-hypertonic lysis is a kind of injury that cells experience when they are exposed to hypertonic media. However, this kind of injury is developed only at the resuspension stage when cells are placed back into the isotonic medium. It has been suggested in the past that the nature of this lysis could be osmotic, when it was suggested that salt loading could occur during hypertonic exposure, thus increasing the intracellular number of salts, leading to cell swelling and possibly lysis upon resuspension. Indeed, cation leaks and salt loading was observed experimentally. However, it was difficult back at these days to explain the salt loading especially because of lack of physiological knowledge. So what I am basically trying to do in this study is to provide a theoretical account based on the modern cell physiology on post-hypertonic lysis of red blood cells. Authors of these studies decades ago assumed that cell plasma membrane are impermeable to ions or cations. Modern cell physiology these views as incorrect assumption. Cell membranes are permeable to ions. Cell membrane contains a wide variety of membrane transport proteins allowing ion movements across plasma membrane. Cells are also experiencing a dynamic equilibrium at normal isotonic conditions when passive dissipative ion fluxes are counteracted by action of ion pumps. This equilibrium allows cells to maintain the normal isotonic cell volume as well as to store energy for performing many cellular functions. Cells also have capacity to increase the ion permeability of plasma membrane substantially, which allows them, besides other things, to accomplish cell volume regulation. Why would cells want to do that? For example, when one changes tonicity of suspension medium, cells may want to adapt to changing conditions by modulating their volume to reduce stress from cell dehydration or cell swelling. Cell physiology recognizes regulatory volume increase and regulatory volume decrease responses. Also, one should differentiate between chronic and acute cell volume regulation. While chronic takes more time to accomplish, generally speaking, acute cell volume regulation is typically accomplished within minutes to tens of minutes. And it is accomplished typically by salt loading or salt loss, leading to volume increase or volume decrease respectively. Based on this consideration, I have recognized that osmotic response of red blood cells is consistent with regulatory volume increase response. So basically in this study, I am hypothesizing that what is actually going on is that cell volume regulation mechanisms are activated during hypertonic exposure. While this is a healthy physiological response, when excessive enough in combination with abrupt dilution, it may induce osmotic lysis. To check whether this hypothesis can account for these observations, I have come up with quantitative descriptions of the physiological state. It's based on the established model developed by other guys. Basically, ion flux equations are solved, ion flux across plasma membrane, which explicitly take into account membrane potential. Regarding the assumed parameters, nothing exotic was assumed. I took normal values from isotonic conditions except the cation conductance, which is increased in accordance with the proposed hypothesis. Regarding the numerical solutions to these equations, on the left side you can see cell response to 100 times increase of sodium conductance. Maybe I should emphasize that the dynamic equilibrium which cell experience at normal conditions, there are considerable driving force for sodium entry into the cells and potassium outflux of the cell. These cells in general are not in chemical equilibrium. So as far as ion channels open, sodium will flow into the cells or potassium will flow out of the cell. So on the left side, when sodium conductance is increased, you get a sodium influx coupled to chlorine ion influx into the red blood cells and eventually volume increase, which exceeds hemolytic volume eventually. On the right side, you can see opposing response when potassium selective ion pathways are open, membrane hyperpolarization occurs, which is associated with potassium loss and chlorine loss from cells and volume decrease. I should emphasize that also in the case of activation of non-selective ion channels, you get eventually cell volume increase, although initially there might be membrane hyperpolarization which induces some volume decrease. I should emphasize the decisive role of membrane potential in all of this. To sum it up, based on these considerations, membrane depolarization leads to solid loading, while membrane hyperpolarization leads to solid loss. Regarding the time aspect of this solid loading by these ion pathways, as expected, more sodium selective the response is, the more quick it is accomplished. Time to reach hemolytic volume is plotted against conductance increase factor. Here I assume normal chlorine conductance, which actually might be even increased in reality, but that's still something that I'm working on. So the time required to reach the hemolytic volume might be actually even reduced than what you see. So far it might appear as an arbitrary play with numbers, but it is actually backed up by experiment. RVI, regulatory volume increase mediators, have been identified to be in red blood cell plasma membrane. This includes especially hypertonicity-induced cation channels and various ion transporters which allow sodium influx. Regarding the cation conductance increase, 3 to 4 orders increase were experimentally observed. So these numbers that I assumed fall within this range. I would like to emphasize, at least in my eyes, rather high explanatory power of this hypothesis when phenomena like beneficial effect of increased cooling rate or gentler dilution might be simply explained when considering first the time aspect of the solid loading, and with the gentler dilution it is that salt, the accumulated salt, might be lost during that process. Also, very exotic observation of freezing-induced swelling of human pulmonary endothelial cells. This hypothesis can explain straightforwardly. Also, I would like to emphasize the abnormal behavior of red blood cells when initially suspended to anisotonic media. They differ in their susceptibility to post-hypertonic lysis. Also, this can be explained by initial involvement of cell volume regulation mechanisms. Also, I would like to highlight the generality of this idea, because post-hypertonic lysis was observed in other cells as well, and cell volume regulation mechanisms are present virtually in all biological cells. Furthermore, I would continue with implication of this hypothesis. If it's true, introduction of ion channel inhibitors into media would or should reduce the post-hypertonic leases, as these would block ion channels, so there would be no soul loading. Also, substitution of sodium with actually non-permanent cations could also help. These predictions are already to some extent confirmed by experiment regarding red blood cells. Chlorpromazine, an ion channel inhibitor, was shown to have protective effect against post-hypertonic leases of human red blood cells. In oocyte cryopreservation, there was observation that choline-based freezing medium shown lesser solution effects injury compared to sodium. To sum it up, I would like to say that osmotic response of human red blood cells corresponds to regulatory volume increase response, which is typically accomplished by gain of sodium chloride. I would like to also emphasize the passive nature of this ion transport mechanism, because these are the most efficient elements of cell volume regulation, allowing the highest ion flux rates. Also, this hypothesis continues the original suggestion of osmotic nature of post-hypertonic leases suggested decades ago. Also, in general, for other cells, I would like to highlight that this might vary depending on cell type. Some cells may be more efficient with cell volume regulation more than others, but basically more efficient regulatory volume increase you get, the higher risk of post-hypertonic leases there is. If you are interested in this, I kind of publish this already as a preprint on bioRxiv. You can visit that. I would like to thank my family, my colleagues, and grant agencies for their support. And there are references for the statement that I used in this presentation, but if you visit the preprint, there are many more of them. Thank you for your attention.
Thank you very much, Ivan. Do we have any questions from the audience?
Really interesting talking about. Thank you for the presentation. RBC is always a challenge, right, among the other type of the cell in the blood, especially for preservation perspective here. One thing I really like to see is now, since you open up something about the electrolyte of the exchange from sodium, potassium, and those, if there is possibility to see what would be the earliest biomarker or something that you can see that the cell become apoptotic if you under those conditions, kind of like a signature. There is no gene, but sometimes they have RBC, they have reticulocyte, they have RNA, or different. I don't know if something is possible.
Erythrocytes can undergo apoptosis-like cell death. It's called eryptosis in their case. I am not sure about markers, but if it was occurring, it would have some specific signs as membrane blebbing or that optically it could have been resolved if apoptosis is occurring. But biomarker, I don't know. I'm not from that area, so I guess I cannot answer that.
Thank you. Do we have any other questions? So I'm just wondering, so we know that in circulation there are younger subpopulations of red cells that are larger and more deformable, and then there are older subpopulations that tend to be much smaller and less deformable. Have you thought about how the post-hypertonic lysis you're talking about here would relate to that, like those different subpopulations?
Yes, I guess there might be differences, and I believe that it was suggested by at least one study that there observed actually bimodal curves in some responses, and they were interpreting it this way, that older erythrocytes behave slightly differently than the young ones, let's say. There could be differences, yes. I don't know. I cannot answer that now, but I will think about this. Thank you for the suggestion.
Thank you.
You mean erythroblasts, erythrocyte, right? The early stage of erythroblasts?
Yeah, because the average lifespan of red cells in circulation is about.
They're close. They're completely different from mature red blood cells.
Yeah, there's other subpopulations because the lifespan of red cells in circulation is about 120 days. So we're talking about mature red cells that are younger versus those that are a little bit older. Yeah.
I've got one maybe final question. So this approach is taken advantage of in many of the modern red cell additive solutions that are using chloride reduced concentrations to try to buffer pH, mainly intracellular pH. But in the early work that Harry Merriman did, where he was actually looking at potassium loading of red cells and saw an improvement in a reduction in post-hypertonic lysis, how does that fit in? How does the whole potassium loading argument fit into some of this?
You mean like a suspension medium was composed of potassium?
Yeah.
Something like this? I cannot do. I guess this from top of my head would need to address this quantitatively, but I think you can get potassium loading also, because, for example, many of these hypertonicity-induced cation channels are actually non-selective, and if they can allow sodium entry, they could also allow potassium entry. So. But you imply that there is reduced post-hypertonic lysis?
Yeah, Harry Merriman in one of his 1970s papers was looking at this in detail.
I need to look it up.
Yeah.
I'm not familiar with that paper, I guess.
All right, thank you. All right, thank you. All right, so our next speaker is joining us virtually. Olena Chernenko is joining us from, I'm not sure actually where Olena is situated right now. Just like to again acknowledge to everyone that our hearts and prayers go out to our colleagues in Ukraine.